Welcome to the deep dive. Our mission today, well, it's a big one. We're tackling how life manages this incredible organizational challenge, packing almost 2 meters of DNA.
2 meters into a space you can barely see the cell nucleus, maybe 5 to 10 micrometers across.
That's just amazing. And it's not just storage, is it? This packing directly controls how our genes work.
Absolutely. It's active organization, how that DNA is folded, coiled, anchored, that determines if a gene can even be read or transcribed. If it's locked away too tightly, well, nothing happens.
So our plan is to start simple. We'll look at viruses, bacteria, see the basic mechanics.
Right, the fundamentals.
Then maybe touch on some unusual eukaryotic structures that gave scientists early clues.
Yeah, the polytene and lampbrush chromosomes, really visual examples.
And finally, dig into the complex, multi-layered packing in our own cells, eukaryotic chromatin. Okay, let's jump in with the simpler stuff.
Okay, so viruses and bacteria, they generally keep it simpler. Often it's a single chromosome, maybe DNA, maybe RNA, sometimes circular. They don't have quite the protein zoo eukaryotes do, but the compaction problem, still massive.
The scale is still wild. Like, wasn't there that lambda phage example, packing 17 millimeters of DNA?
Into a head less than 1/10 of a millimeter, exactly. And bacterial chromosomes, like E. coli's, are huge, to 1,200 micrometers long. That all has to fit into the nucleoid.
So how do they manage that initial crunch?
They use associated proteins, sometimes called histone-like proteins. HU and HNS are common ones in bacteria. They're positively charged.
So they stick to the negatively charged DNA backbone.
Precisely. Simple electrostatic interaction helps fold and bend the DNA. But a really key mechanism, especially for circular DNA, is supercoiling.
Right, supercoiling. Crucial for compaction. How can we picture that without, waving our hands around?
Okay, imagine a rubber band, a circular one. It's relaxed. Now hold one part and twist the other end. Underwind it.
Okay, yeah, it gets strained.
Exactly. And what happens? The whole thing wants to coil up on itself to relieve that strain. That's basically DNA supercoiling. If you take a relaxed circle of DNA with a certain number of natural twists, that's its linking number, and you force it to underwind,
it buckles.
It buckles into tight, compact twists called supercoils. For DNA, underwinding typically leads to negative supercoils, which compacts it.
And we see that in viruses like SV40. You mentioned it's underwound.
By about 25 turns, resulting in 25 negative supercoils. Huge compaction effect. And molecules that only differ by this linking number, by the number of supercoils, are called topoisomers.
So the cell needs a way to manage this twisting and untwisting.
It does. That's where topoisomerases come in. These enzymes are the controllers. Type A enzymes tend to relax the DNA, removing supercoil.
Well, you breathe a bit.
Sort of, yeah. But type 2 topoisomerases, they can actually introduce negative supercoils. They do this rather drastically, cutting both DNA strands, passing another part of the DNA through the break, and then sealing it back up.
Wow, molecular surgery to control shape.
Pretty much. And this isn't just prokaryotes. Eukaryotes use topoisomerases too, because even though their DNA isn't always circular, it's anchored in loops, creating topologically constrained domains where supercoiling matters.
Okay, that makes sense. That fundamental idea of structural control bridges us to eukaryotes, where, as you said, some early observations were key.
That's right. Long before we could sequence genomes or visualize single molecules, scientists got clues from these really unusual giant chromosomes. Polytene and lampbrush chromosomes.
Polytene chromosomes. Those are the ones in things like fruit fly salivary glands.
Yep, insect larvae often have them. They're weird because the homologous chromosomes pair up and then replicate over and over, maybe thousands of times, but the strands don't separate.
So you get these massive, thick chromosomes with visible bands. Chromomeres, right?
Exactly. And the breakthrough was noticing these localized areas where the bands seemed to swell or puff out. Puffs, they called them.
And the puffs meant?
Gene activity. Researchers used radioactive RNA precursors, the building blocks of RNA, and showed they were incorporated heavily in those puffed regions. It was direct visual proof. Structural change, this uncoiling, equals active transcription.
A direct link between form and function. What about lampbrush chromosomes?
Similar idea, different context. Found often in vertebrate oocytes during meiosis. They look, well, like an old-fashioned lampbrush used to clean chimneys.
A central axis with loops sticking out.
Pairs of lateral loops, yeah. They're essentially decondensed meiotic chromosomes. And again, experiments showed those loops were sites of intense RNA synthesis.
So both were like natural experiments, showing that chromosomes have to physically change shape, to decondense, to allow genes to be expressed.
Exactly. They set the stage for understanding chromatin in all eukaryotic cells.
Which brings us to the standard state. Chromatin. That dispersed DNA-protein complex in the nucleus during interphase. But when the cell divides, it has to condense dramatically.
By about 10,000-fold in length into those visible mitotic chromosomes. It's an incredible packaging process.
And the key players here are the proteins, especially the histones.
Right. Chromatin is primarily DNA complexed with histones. There are 4 core histones: H2A, H2B, H3, and H4. They are packed with positively charged amino acids, lysine and arginine.
So they naturally bind very tightly to the negatively charged phosphate backbone of DNA.
It's a fundamental interaction, and it leads to the basic structural unit of chromatin.
The nucleosome. The beads on a string. How was that figured out?
It came from digestion experiments. When chromatin was treated with enzymes that cut DNA, the DNA wasn't just randomly chewed up. It was cut into fragments of roughly 200 base pairs, or multiples of 200.
Suggesting a repeating protected unit.
Exactly. And then electron microscopy provided the visual. These little beads, the nucleosomes, connected by threads of linker DNA. The bead itself, the core particle, is about 147 base pairs of DNA wrapped around an octamer.
Octamer meaning 8 proteins.
Yes, 2 copies each of the 4 core histones, H2A, H2B, H3, and H4. The DNA makes about 1.7 left-handed turns around this protein core.
And there's a 5th histone, H1.
Right. H1 sits outside the core particle, associated with the linker DNA, and helps lock the DNA wrap in place. It's crucial for the next level of packing.
So this nucleosome formation, the beads on a string, that's the first level. How much compaction does that achieve?
It gives about a 3-fold reduction in length, maybe a bit more. Creates an 11 nanometer diameter fiber.
3-fold. Good start, but nowhere near 10,000-fold. What's next?
The 11 nanometer fiber coiled up, often described as a solenoid structure, though the exact geometry is still debated a bit. This forms a thicker fiber, about 30 nanometers in diameter.
The 30 nanometer fiber. And H1 is important for this step.
Very important. It helps stabilize this higher order coiling. This step gives another, say, 6-fold compaction, so now we're getting somewhere.
From 11 nanometers to 30 nanometers. Then what?
Then that 30 nanometer fiber gets organized into large loops or domains. These loops, maybe 300 nanometers wide in total structure, seem to be anchored to a protein scaffold within the chromosome.
Looping the rope, essentially.
Kind of, yeah. And then these loop domains fold and coil further to form the compact chromatid arms we see in metaphase chromosomes, which are about 700 nanometers wide each, making the whole chromosome about 1,400 nanometers across. It's this hierarchy of folding.
DNA helix, nucleosomes, 30 nanometer fiber, loops, chromatid. Incredible layers of organization.
But what's crucial to remember is that this isn't static. It has to be dynamic.
Right, because if it's packed that tightly, how does anything get transcribed or replicated?
Exactly. The machinery, the enzymes like RNA polymerase can't access DNA when it's locked up in that 30 nanometer fiber or higher structures. So the cell needs mechanisms for chromatin remodeling.
Unpacking it locally when needed.
Precisely. Relaxing the structure to expose specific DNA sequences.
And a key control point seems to be the histone tails, right? Those bits sticking out from the nucleosome core.
Yes. Those tails are accessible and are targets for all sorts of chemical modifications. They act like little switches.
What's the most well-understood modification?
Probably acetylation. Enzymes called histone acetyltransferases, or HATs, add an acetyl group to lysine residues on the tails. Lysine is positively charged.
And acetylation neutralizes that positive charge.
Right, which weakens the grip between the histone tail and the negatively charged DNA. It loosens things up. And this is strongly correlated with gene activation. Open chromatin, active genes.
Is there direct evidence for that link?
Oh, yes. A classic example is the inactive X chromosome in female mammals, the Barr body. It's known to be heavily underacetylated compared to the active X chromosome. Low acetylation, condensed, inactive chromatin.
Makes sense. What other modifications happen on those tails?
There's methylation adding methyl groups to lysines and arginines. This one's more complex. Methylation can be associated with either activation or repression, depending on which specific amino acid gets methylated and how many methyl groups are added.
So context matters for methylation.
Very much so. And then there's phosphorylation, adding negatively charged phosphate groups, often to serines. This modification seems more involved in the large-scale condensation cycle, like during mitosis.
Okay, so histone modifications mainly affect accessibility. But you mentioned methylation. Is that different from methylating the DNA itself?
Ah, yes, critical distinction. Histone methylation modifies the protein. DNA methylation modifies the DNA base itself, specifically cytosine, often found in CpG sequences. And unlike histone methylation, which can go either way, DNA methylation is almost always associated with gene silencing. It's a repressive mark directly on the gene sequence.
Got it. So based on this condensation state and activity, chromatin gets broadly classified, right?
Into two main types, euchromatin and heterochromatin.
Euchromatin is the lighter staining stuff.
Yeah.
More open.
Generally, yes. It's less condensed, stains less intensely, and it's where most of the active genes reside. It replicates earlier in the S phase of the cell cycle.
And heterochromatin is the opposite.
Right. It remains highly condensed throughout the cell cycle, even during interphase. Stains darkly. It's largely genetically inactive. Either it lacks genes or the genes within it are repressed, and it tends to replicate late in S phase.
Where do we typically find heterochromatin?
It's crucial for chromosome structure. The centromeres and telomeres are made of heterochromatin. Large portions of some chromosomes, like the mammalian Y chromosome, are heterochromatic. And the inactive bar body X chromosome is another classic example.
So it's structural, but also repressive. What about that position effect you mentioned earlier?
That's a fascinating phenomenon. It shows just how powerful the heterochromatic environment is. If you experimentally move a normally active gene from a euchromatic region right next to a block of heterochromatin.
It can get shut down.
Often, yes. The silencing effect of the heterochromatin can spread into the newly adjacent gene, turning it off simply because of its new location. It highlights that chromatin environment is critical for gene expression.
And to visualize these regions and chromosomes overall, especially during mitosis, cytogeneticists use banding techniques.
Yes. C-banding is one method that specifically stains the constitutive heterochromatin, particularly around the centromeres.
But the most common one is G-banding.
G-banding is the workhorse for clinical cytogenetics and research. It involves a mild protease treatment, like trypsin, followed by GIMSA staining. This produces a unique pattern of alternating light and dark bands along the entire length of each chromosome.
Like a barcode for each chromosome.
Exactly like a barcode. It allows researchers to unambiguously identify each chromosome pair and, crucially, detect structural abnormalities like deletions, duplications, or translocations between chromosomes.
Okay, so we've covered structure and its relation to activity. Now let's shift focus slightly to the DNA sequence itself. Beyond the genes, what else is in there? Especially in eukaryotes.
Well, that's where things get really interesting and, frankly, surprising. The eukaryotic genome isn't mostly genes. A huge fraction is made up of repetitive DNA sequences.
Repetitive DNA, how's that categorized?
Usually based on how repetitive it is and how the repeats are arranged. The most extreme is highly repetitive DNA, sometimes called satellite DNA.
Why satellite?
Because when you centrifuge genomic DNA in a density gradient, these highly repetitive sequences often form separate satellite bands because their base composition, and thus density, can differ slightly from the bulk of the DNA.
And what are these sequences like?
They are usually very short sequences, just a few base pairs to maybe a couple hundred, repeated thousands or even millions of times, arranged end-to-end in tandem arrays.
Located where?
Primarily in heterochromatic regions, especially the centromeres and telomeres. Their function seems mostly structural, not informational.
Okay, that's highly repetitive. What about middle or moderately repetitive DNA?
This is a much broader category. It includes some functional genes that exist in multiple copies because the cell needs large amounts of their products. Ribosomal RNA genes are a classic example.
Makes sense to have backups for essentials.
Exactly. But this category also includes vast amounts of non-coding repetitive sequences. These fall into two main patterns.
Which are?
Tandem repeats, like satellite DNA, but usually in shorter stretches. This includes things like VNTRs, variable number tandem repeats, or mini-satellites, and STRs, short tandem repeats, or microsatellites.
Those sound familiar. Forensics, DNA fingerprinting.
Precisely. The number of repeats in these tandem arrays varies greatly between individuals, making them incredibly useful as genetic markers. Microsatellites, like simple CA repeats, are scattered throughout the genome.
Okay, so tandem repeats. What's the other pattern for middle repetitive DNA?
Interspersed repeats. These aren't clustered together, they're scattered individually throughout the genome. And many of them are actually mobile genetic elements or remnants of them, often called transposons or retrotransposons.
They can move around.
Or at least they originated from elements that could. Many transpose via an RNA intermediate using an enzyme called reverse transcriptase to make a DNA copy that then inserts somewhere else.
So these are the signs and lines I've heard about.
Exactly. Signs are short interspersed elements, typically less than 500 base pairs. The ALU family is the most famous human. There are hundreds of thousands of ALU sequences, making up something like 5% or more of our entire genome.
Wow, and lines.
Long interspersed elements, these can be much larger, up to 6 kilobases. The L1 family is the predominant line in humans.
Oh.
Both signs and lines have proliferated enormously during mammalian evolution.
So if you add up all this repetitive stuff, the highly repetitive satellite DNA, the middle repetitive VNTRs, STRs, signs, lines, what does that leave for actual genes?
That's the truly astonishing part. When you sum up the highly and middle repetitive DNA, it accounts for roughly 50% of the entire human genome, half.
Half our DNA is repetitive elements.
At least. And then you have other non-coding sequences, introns within genes, regulatory regions, pseudogenes.
Which brings us to the final stark statistic.
Only about 2%, maybe even less, of the total human genome sequence actually codes for proteins. The 20,000 or so functional genes we have are embedded within this vast sea of non-coding and repetitive DNA.
98% non-coding. That just reframes everything, doesn't it? It's not just about the genes. It's about managing this enormous, complex landscape.
Absolutely. We've gone from the simple, elegant supercoiling in bacteria
Through the visible drama of puffs and loops.
to the intricate, chemically tagged, hierarchical folding of our own chromatin. And realize that controlling access to that tiny 2% of coding sequence is perhaps the most fundamental challenge.
It really puts gene regulation in a new light, which leads to a final thought for you, our listener, to ponder.
We know that this default state is compaction, tight folding. We know chemical tags like acetylation act as signals to open it up.
So the question is, how does the cell achieve the exquisite specificity? How does it ensure that precisely the right genes within that tiny 2% are unpacked and transcribed only when and where they are needed?
What are the higher-level cues? How does the cell navigate this vast non-coding landscape to pinpoint the right targets against that massive backdrop of condensed chromatin?
Something to think about. It's a central question in biology today.
It really is. Well, thank you for joining us on this deep dive into DNA's packaging problem.
We hope untangling some of this complexity helps you tackle your own challenges in genetics. Thanks for listening.